Hi friends, today we are dealing with the topic genetics and introduction. Firstly, what is genetics? Genetics is a study of structure and function of gene and genetic variation. Transmission of genes from one generation to the next is termed as heredity. In genetics, we study about these two terms, gene and heredity. By the end of the session, you should be able to learn the basic concepts like genes and alleles, traits and characters, different branches of genetics, DNA and chromosome, homozygous, heterozygous and hemizygous condition, genotype and phenotype, wild type and mutant type, dominant and recessive, and finally the genetic symbols. Next, we discuss about different branches of genetics. First, we look into transmission genetics. This branch of genetics deals with the basic principles of genetics and how traits are being transmitted or inherited. Example for this branch is the pattern of trait transmission in a human pedigree. Next is the molecular genetics. This branch of genetics deals with the chemical nature of genes and how it is encoded, replicated, and expressed. In molecular genetics, we study about chemical nature of gene, which means the nucleotide sequence of the DNA, and how it is encoded, how it is replicated, and how it is expressed by the process called transcription and translation. Example for this branch is the molecular events involved in the control of cell division. Next is the population genetics. This branch of genetics deals with the study of genetic composition of populations and how populations' collective group of genes changes with time. Example for this branch is the frequency of a disease-causing gene in human population. Finally, the fourth branch, that is quantitative genetics, deals with the study of genetic analysis of complex characters which are influenced by many genes. Example for this branch is the fruit weight in agricultural plants. In agricultural plants, the fruit weight which is controlled by or which is influenced by many genes or which is a polygenic interaction, the genetic analysis of such complex characters are included in quantitative genetics. What is a gene or a Mendelian factor? A gene is a genetic factor that helps to determine a particular trait which is passed from parents to offspring. In molecular terms, a gene is a nucleotide sequence in DNA that specifies a polypeptide or RNA. In Mendelian terms, a gene is a trait, whereas in molecular terms, a gene is a nucleotide sequence in DNA that specifies a polypeptide or RNA. What is the difference between character and trait which we use all along genetic concept? A character is an observable physical feature which results from a gene action and which is normally transmitted from one generation to the next. Example for character is flower color. Flower color is considered to be a character which is an observable physical feature which results from a gene action, whereas a trait is a particular form of a character. Example for traits are red, white, pink, etc. The different forms of colors like red, white, pink, these all are considered to be the traits of a character flower color. There are particular forms of character and that particular forms of character are termed as traits. Next, let us move on to the detailed study of a chromosome. What is a chromosome? A chromosome is a linear structure composed of a single DNA molecule which is complexed with specific proteins called histones. Each eukaryotic species has a characteristic number of chromosomes. Example, humans contains 46 chromosomes, whereas the model organism Drosophila contains 8 chromosomes. Look into this picture shown here. Chromosome is a DNA double helical structure which is wrapped around by histone proteins and which undergoes various modifications like chromatin fiber as shown in the figure and finally attains a structure called chromosome which we discussed earlier as a linear structure composed of a single DNA molecule and which is complex with specific proteins called histones. Next, we look into deoxyribonucleic acid or DNA. A DNA is a polymeric molecule consisting of deoxyribonucleotide which are arranged in a double-stranded helical form and is a genetic material in almost all organisms. This picture shows the double helical structure of DNA along with the sugar phosphate backbone. Next, let us discuss about different parts of a chromosome. This picture shows a single chromosome with its parts. Let us look into one by one. Firstly, centromere, the constricted region of the chromosome to which spindle fibers attach. Spindle fibers are attached to a specialized multi-protein complex called kinetochore at the centromere. Chromatid, these are one of the two visibly distinct replicated copies of chromosome. Telomeres are a specific set of sequences at the end of a linear chromosome that stabilizes the chromosome. Origin of replication, these are a specific region in DNA where the double helix unwind and replication starts. What is the difference between duplicated and unduplicated chromosome? This picture clearly defines the difference between these two terms. An unduplicated chromosome contains single chromatid and is observed before replication, whereas a duplicated chromosome contains two chromatids, that is sister chromatids, which are formed after replication of chromosomes. Look into the box shown here. This is a shortcut to find out the number of chromosome and number of DNA inside a cell. Number of chromosomes can be calculated by counting the number of centromeres. For example, in humans there are 46 number of chromosomes. These are calculated by counting the number of centromeres. And the number of DNA can be calculated by counting the number of sister chromatids inside a cell. In humans, there are 46 number of DNA before replication and 92 number of DNA after replication. This can be calculated by counting the number of sister chromatids inside the cell. What are homologous chromosomes? Homologous chromosomes are the chromosomes that have the same structure, size, and carry genetic information for the same set of hereditary characteristics like hair color, skin color, etc. In a pair of homologous chromosome, one is inherited from the male parent and the other from the female parent. Look into the picture shown here. Here, the blue represents the chromosome from the father and pink represents the chromosome from mother. The chromosome with one chromatid undergo duplication and forms chromosome with two chromatid called sister chromatids. In the same way, the chromosome which is in pink color, that is from the mother, also undergoes duplication and forms chromosome with two chromatids called sister chromatids. These two chromosomes from father and mother forms homologous chromosomes. What are genes, locus, and alleles? What is a gene? A gene is a nucleated sequence in DNA or chromosome. Locus refers to the position of a gene on a genetic map. These are the specific space on a chromosome where the gene is located. Alleles are one of the two or more alternative forms of a single gene that can exist at the same locus. All alleles of a gene determine the same hereditary trait, example, seed color, but each allelic composition has its own nucleotide sequence. Look into the picture shown here. This picture represents a homologous chromosome, that is, one from father and one from mother. Here, the colors pink, orange, blue, green, yellow represents the gene locus, and small r, capital P, capital P, capital A, small b, small c, and capital C represents the allelic composition that are present in specific gene locus. Here, the capital A refers to the homozygous dominant condition. Small b refers to homozygous recessive condition, and capital C termed as heterozygous condition. What are homozygous, heterozygous, and hemizygous condition? What is a homozygous condition? For a diploid organism having the same alleles at both locus, that is, if same type of alleles are present in both locus, it is referred as homozygous condition. If alleles are same, the gametes produced will be single type or same type. For example, the genotype small a means both the alleles are small a, and the gamete produced is a single type, that is small a. What is heterozygous condition? For a diploid organism having different alleles at each locus and therefore produces two types of gametes. For example, the genotype capital A composed of two different alleles, capital A and small a. Therefore, they are heterozygous in nature and produces two different types of gametes, capital A and small a. What is a hemizygous condition? Possessing only one copy of gene or one copy of allele in a diploid cell. Usually, genes on the X chromosome in males with the XY genotype. As we all know, males are with XY genotype, and if a gene is present on the X chromosome, it will be present only in single copy, and such condition is referred as hemizygous condition. What is genotype and phenotype? Genotype refers to the complete genetic makeup or allele composition of an organism. Example, capital A small a refers to the allele composition which represents the genotype, whereas phenotype refers to the observable characteristics of an organism that are produced by the genotype and its interaction with environment. Example, yellow, green, etc. What is wild type and mutant type? Wild type describes an allele or gene that is designated as a standard or normal for an organism and is the most prevalent in the wild population of an organism. That is, wild type is considered to be the standard, normal, most commonly seen, most prevalently seen in the population. For example, if tall phenotype is the common, it is considered to be the wild type, and the dwarf phenotype is the mutant type. That is, if tall phenotype is the commonly seen, most prevalently seen in a population, it is considered to be the wild type, whereas the dwarf phenotype is considered to be the mutant type. Then what is mutant type? Any form of allele or gene that differs from the wild type allele or gene. Wild type, as we already discussed, is the most standard, normal, commonly seen, most prevalently seen in a population. Anything which is different from wild type is considered to be mutant type. Mutant alleles can be dominant or recessive to the wild type alleles. This is a very important point to be noted. Most of us think that mutant alleles can always be recessive. This is not the case. Mutant alleles can be dominant or recessive to the wild type alleles. What is dominant and recessive condition? Dominance refers to the condition in which an allele is phenotypically expressed when one or both copies are present. If a single allele or if two alleles are present and when it is phenotypically expressed, then the condition is dominant. In dominant condition, the phenotype of the heterozygote is same as that of the homozygote. In genetics, the dominant allele is normally represented by uppercase letters like capital A, capital B, capital D, capital R, etc. Example, capital D represents the tall allele. Then what is recessiveness? The condition in which an allele is phenotypically expressed only when two copies are present. In genetics, the recessive allele is normally represented by lowercase letters like small a, small b, small d, small r, etc. Example, small d represents dwarf allele. Next, let us study how to represent gene symbols. In genetics, there is a conventional method representing gene symbols. First one is a Mendelian terminology which is used for plants and most animals. We already discussed that in genetics, the dominant allele is represented by uppercase letter, for example, capital D, whereas recessive allele is represented by lowercase letter, for example, small letter d. Which letter to be chosen is determined by the mutant phenotype. For example, tall and dwarf are the two traits of a character plant height. If dwarf is considered to be the mutant phenotype, the gene symbol taken for representing the respective traits are denoted by the letter capital D or small letter d. That is, for the trait tallness, which is dominant, is represented by capital D and not by capital T. And for the recessive trait dwarfness is represented by small letter d instead of small letter t. That is, letter for representing gene symbol is determined by the mutant phenotype. Here, dwarfness is a mutant phenotype. Therefore, the letter for representing the trait is taken from the first letter of mutant phenotype, dwarfness. Next method for representing gene symbol is Drosophila symbolism, which is commonly used in genetics today. The symbol plus sign indicates the dominant type allele and lowercase letter represents the recessive allele. Letters are used based on mutant phenotype. As we already discussed, letter to be chosen is determined by the mutant phenotype. For example, if red eye color is dominant over white eye color, the red allele or the red eye color which is dominant is represented as plus sign. And white allele or white eye color which is recessive is represented by letter W. Here, white is the phenotype produced by the mutant allele. Here, the letter W is determined by the mutant phenotype. In this example, white allele or white eye color is considered to be the mutant phenotype. Therefore, the letter chosen for representing the trait is W. And small letter w indicates or denotes white eye color is recessive in nature. If there is one more trait in the same organism in which a mutant phenotype starts with the same letter, small letter w, for example, the trait wingless, the representation of the allele is small w and small g. That is, one more letter is added to this. Next is the representation of multiple alleles. For multiple alleles, the allele is represented by superscript to a letter, that is, the letter commonly represents the main character. Here, the dominant allele is represented by plus symbol and recessive by lowercase letter. Example, coat color in rabbits which have 4 alleles, that is, agouti which is represented by C+, Himalaya which is represented by CH, Chinchilla by CCH and albino by C. Here, C represents the main character, that is coat color, whereas plus, H, CH and C represents the multiple alleles, which are represented by superscript to a letter, that is, the letter which is commonly represents the main character. Here, C represents the main character, coat color. Next, how we can represent the X-linked traits. For X-linked traits also, the allele is represented by superscript to X chromosome. Here also, dominant allele is represented by plus symbol and recessive by lowercase letter. Example, in an X-linked trait, that is red-green color blindness, X superscript to C represents the recessive allele that causes color blindness, and X superscript to plus sign represents the normal dominant allele. Remember that for X-linked trait and multiple alleles, the allele should be represented superscript to X chromosome in case of X-linked trait and to a letter which is represented by the main character in case of multiple alleles. That's all about today's class. Next unit, we are discussing about Mendelian genetics. In Mendelian genetics, we will cover the topics like genetic process, Mendel's law of genetics, an initiative from Life Science Academy. Thank you.